Welcome to cell biology. Have you ever wondered how a cell, which is essentially a microscopic fortress, manages to take in the nutrients it needs and get rid of the waste it produces? Today, we are going to explore the fascinating world of transport across membranes. We will learn exactly how molecules travel in and out of cells, ensuring that our bodies function smoothly at the most fundamental level. Let us dive right in. To understand our first topic, imagine being in a crowded room where everyone is packed tightly into one single corner. Naturally, people will start spreading out to the empty spaces until the room is evenly filled, simply because it is more comfortable. This natural tendency to spread out without needing any extra energy is exactly how passive transport works in a biological cell. The cell membrane acts like a doorway, and molecules simply wander from where it is highly crowded to where it is relatively empty. You do not need to push them. They move entirely on their own due to their natural kinetic energy. This process is vital because it allows cells to effortlessly take in resources like oxygen without wasting precious energy. Now that we understand what passive transport is, let us look at how it actually happens across the cell membrane. There are two main ways molecules can passively enter or exit a cell. The first is simple diffusion. This happens when small, nonpolar molecules, like oxygen and carbon dioxide, slip directly through the lipid bilayer of the membrane, almost like ghosts passing through a wall. However, not all molecules can do this. Larger or charged molecules, such as glucose or sodium ions, cannot easily pass through the water-fearing core of the membrane. For these, the cell uses facilitated diffusion. This process relies on specific transmembrane proteins that act as dedicated tunnels or doorways, allowing these larger molecules to safely cross without using any cellular energy. This concept of molecules spreading out naturally leads us to a mathematical way to measure it, known as Fick's first law of diffusion. Conceptually, this formula tells us exactly how fast molecules are moving across a given area of the membrane. Let us break down the symbols. J represents the diffusion flux, which is the amount of substance moving through a unit area per unit time. D stands for the diffusion coefficient, telling us how easily a specific molecule moves through the medium. Delta C is the difference in concentration across the membrane, and delta X is the membrane's thickness. The negative sign is crucial. It indicates that the flow is strictly down the concentration gradient, from high to low. If the concentration difference increases, the flux increases proportionally, meaning molecules move much faster. Before we move on, let us see this formula in action with a concrete example. Imagine a cell membrane with a thickness of 5 × 10 to the power of -9 meters. The concentration of oxygen outside the cell is 50 moles per cubic meter, and inside it is 10 moles per cubic meter. The diffusion coefficient is 1 × 10 to the power of -11 square meters per second. We need to find the diffusion flux of oxygen into the cell. Because the outside concentration is higher, oxygen will naturally flow inward. Our first step is to calculate the concentration difference, delta C, by subtracting the outside concentration from the inside concentration. This gives us -40 moles per cubic meter. The negative value confirms the gradient points inward. Now that we have our concentration difference, let us complete the calculation. We substitute all our known values into Fick's first law. We place our calculated -40 in the numerator and the membrane thickness in the denominator, multiplying by the negative diffusion coefficient. Notice how the negative sign from the formula and the negative sign from our concentration difference cancel each other out. This ensures our final answer makes physical sense as a positive inward flow. After dividing and multiplying, we arrive at our final answer. 8 times 10 to the power of -2 moles per square meter per second. Physically, this means that every second, a steady stream of oxygen molecules successfully crosses every square meter of the cell membrane, keeping the cell oxygenated and alive. Having calculated the flux, let us visualize exactly what is happening at the microscopic level. On the screen, you are looking at a cross-section of a cell membrane, which is the double layer of lipids. Notice the top area represents the outside of the cell, where there is a high concentration of molecules, and the bottom is the inside, with a low concentration. On the left, you can see simple diffusion. Small blue oxygen molecules are slipping right between the lipids. On the right, we see facilitated diffusion. Here, larger green glucose molecules cannot pass through the lipids directly. Instead, they are traveling through that large purple structure, which is a transmembrane protein acting as a dedicated channel. The large downward arrow reminds us that both processes are driven by the natural concentration gradient. While passive transport is effortless, cells often face a different challenge, which brings us to active transport. Imagine trying to push your way into that same crowded corner of the room when you are currently standing in the empty space. People are packed tightly and pushing against you, so you have to use your own physical energy and muscle to force your way in. This challenging scenario is exactly what active transport is in a biological system. The cell frequently needs to move molecules against their natural flow, from an empty area into a highly crowded area. Because this process moves substances against their concentration gradient, it is thermodynamically unfavorable and cannot happen spontaneously. To achieve this uphill battle, the cell must spend its own hard-earned energy, typically in the form of ATP, using specialized protein pumps. Now that we know active transport requires energy, let us explore the two main ways cells apply this energy. The first is primary active transport. This method directly burns chemical energy, usually ATP, to power specialized protein pumps. A famous example is the sodium-potassium pump, which is absolutely vital for human nerve cells. It maintains a high concentration of potassium inside and sodium outside, creating the specific imbalance necessary for firing electrical signals. Without it, you could not think or move. The second method is secondary active transport. Instead of using ATP directly, it cleverly uses the energy stored in an electrochemical gradient, which was previously created by primary active transport, to drag other substances uphill against their will. Both mechanisms are essential for keeping the cell's internal environment strictly controlled. Since active transport requires energy, we need a way to calculate exactly how much energy is spent. This brings us to the Gibbs free energy of transport equation for an uncharged solute. Conceptually, this formula tells us the thermodynamic cost of moving molecules against their will. Let us look at the symbols. Delta G represents the change in Gibbs free energy, telling us the energy required per mole of solute. R is the universal gas constant, and T is the absolute temperature, measured in Kelvin. C in and C out represent the concentrations of the solute inside and outside the cell. The natural logarithm compares these two concentrations. If the inside concentration is greater than the outside, the logarithm is positive. This makes delta G positive, mathematically proving that energy must be added to force the molecules inside. Let us apply this thermodynamic formula to a real cellular problem. Imagine a cell needs to pump an uncharged nutrient molecule into its cytoplasm. The concentration of this nutrient outside the cell is just 1 millimolar, but inside it is already much higher at 100 millimolar. The body temperature is 310 Kelvin, and the ideal gas constant is 8.314 joules per mole Kelvin. We need to calculate the free energy change required to transport 1 mole of this nutrient. Because the cell is moving the nutrient from a low concentration to a high concentration, we know this is an uphill battle. Our first step is to calculate the concentration ratio by dividing the inside concentration by the outside concentration. This gives us a ratio of 100. With our ratio calculated, we can now find the total energy requirement. We substitute the gas constant, the temperature, and our ratio of 100 into the Gibbs free energy equation. Next, we evaluate the natural logarithm of 100, which is approximately 4.605. We are now ready to multiply these three values together. This multiplication converts our abstract ratio into a concrete energy value. After multiplying, we get a final result of approximately 11,867 joules per mole. Notice that our final answer is a positive number. Physically, this positive value means that the process is not spontaneous. The cell must actively spend at least this much energy, likely by burning ATP, to successfully transport the nutrient inside. To fully grasp this energy-demanding process, let us look at a diagram of a primary active transport pump. Here, you see the cell membrane with a large transmembrane protein pump embedded in it. Notice the environments. The extracellular fluid at the top has high sodium and low potassium, while the intracellular fluid at the bottom has low sodium and high potassium. Follow the red arrows. The pump is forcing sodium ions out of the cell, moving them into an area that is already crowded with sodium. Simultaneously, the blue arrows show potassium being forced inside. This double uphill movement is impossible without energy. That is why you see the yellow starburst at the bottom, representing ATP being broken down into ADP and phosphate. This energy release is what physically drives the pump. We have covered a lot of ground today, so let us summarize the most important ideas. First, we learned that passive transport is the cell's free ride, moving molecules down their concentration gradients without spending any energy. We used Fick's first law to mathematically calculate the rate of this spontaneous diffusion. Second, we explored active transport, which is the cell's heavy lifting mechanism. It uses energy, like ATP, to force molecules against their natural gradients. We then used the Gibbs free energy equation to calculate exactly how much energy this uphill battle costs. Together, these two contrasting processes strictly control what goes in and out of the cell, maintaining the delicate balance required for life. You now have a solid understanding of membrane transport. Great job!